Greetings, dear participants. Myself, Dr. A. Jayaprakash, Assistant Professor, UG and PhD Head, PG and Research Department of Biochemistry, Saidot College Autonomous, Tirupattur District, Tamil Nadu, India. Today, in this lecture, we are going to see about structure, chemical composition, and functions of ribosomes. What is a ribosome? Ribosome is a complex molecular machine found inside all living cells that produces proteins from amino acids during a process called as protein synthesis or protein translation. This process of protein synthesis is a primary function which is performed by all the living cells. Ribosomes, they are specialized cellular organelles that are found both in prokaryotic as well as eukaryotic cells. Every living cells it requires ribosomes for protein synthesis. This cell organelle also functions by binding to a messenger ribonucleic acid, that is mRNA, and decoding the information carried out by nucleotide sequence of the mRNA. They transfer RNAs, that is tRNA, comprising of amino acids and enter into the ribosome at the acceptor site. Once it gets bounded up, it adds amino acid to the growing protein chain on tRNA. So, ribosome is a very complex molecular machine which mainly involves in the protein synthesis. Coming to the structure of ribosome contains two subunits, the large subunit and smaller subunit. Smaller subunit, it contains this mRNA which contains the codon which code for a particular amino acid in tRNA. This tRNA, it goes on adding the activated amino acid chain in order to get a protein being synthesized. So ribosome, it contains a large subunit as well as a smaller subunit where the protein biosynthesis mainly takes place in ribosomes. Some of the characteristic features of ribosomes are spherical in shape. Their size remains constant. The ribosomes of prokaryotes are smaller, that is 150 angstrom, and that of eukaryotes are larger, which is 250 angstrom in diameter. Each ribosome consists of two subunits, namely a larger subunit and a smaller subunit. The subunits occur separately in the cytoplasm. They occur separately, so when they join together, they form ribosomes only at the time of protein synthesis. In other words, in the rest of the time, these are not joined with each other. They occur separately in the cytoplasm, but they join together to form ribosomes only at the time of protein synthesis. As already I mentioned in the structure, the small subunit holds the mRNA during protein synthesis, whereas the large subunit has two slots, namely P site and the A site. What is the P site? P site carries tRNA that contains the polypeptide chain where amino acids are being linked to form a protein molecule. Whereas the A site carries a tRNA containing activated amino acids. Once the amino acids gets activated, finally it forms to a particular protein molecule that is being transported from the cell. According to the size and sedimentation coefficient, ribosomes are classified into two major types, 70S ribosome and 80S ribosomes. What are 70S ribosomes? The 70S ribosomes are found in prokaryotes. They are smaller in size and it has a sedimentation coefficient of 70S. It has a molecular weight of 3 into 10 to the power of 6 daltons. It is composed of ribosomal RNA and proteins. The 70S ribosome is made up of two subunits, namely a larger 50S subunit and a smaller 30S subunit. This 50S subunit is further classified into 23S rRNA, 5S rRNA, and about it contains 30 different proteins. The 30S subunit has 16S RNA and it contains 20 different proteins. These are the characteristic features of 70S ribosomes. Coming to the 80S ribosomes, they are found in eukaryotic cellular organization. They are larger in size and has a sedimentation coefficient of 80S. It has a molecular weight of 5 into 10 to the power of 6 daltons. It is composed of ribosomal RNA and proteins. The ATS ribosome is made up of two subunits, namely the largest subunit is 60S and the smaller subunit is 40S. The 60S subunit is composed of 20S are ribosomal RNA, 5.S are ribosomal RNA, 5S ribosomal RNA and about 50 different protein molecules. The 40S subunit has 18S rRNA and 30 different ribosomal proteins. These are the characteristic features of ATS ribosomes, which is found mainly in the eukaryotic organization. Coming to the difference between 70S and 80S ribosomes, it occurs, 70S ribosomes mainly occur in prokaryotes, whereas 80S ribosomes occur mainly in eukaryotes. The size is very small. The 70S ribosome is very small. It is around 18 nanometer, whereas 80S ribosome is large and it is around 22 nanometer. The sedimentation coefficient is 70S for 70S ribosomes and 80S for 80S ribosomes. The biosynthesis that takes place in 70S ribosome in cytoplasm, whereas 80S ribosomes, the biosynthesis takes place in nucleolus. The two important subunit of 70S ribosome is 50S and 30S, whereas in 80S ribosomes it is 60S and 40S. Okay, what are the different types of ribosomal RNA? There are three types in 70S ribosomes, 5S, 16S and 23S, whereas 80S ribosomes contain four types, 5S, 5.8S, 18S and 28S. 70S ribosomes contain around 50 ribosomal proteins, whereas 80S ribosomes contains around 80 ribosomal proteins. The molecular weight of 70S ribosome is 3 into 10 to the power of 6 daltons, whereas 80S ribosomes is 5 into 10 to the power of 6 daltons. The RNA protein ratio in 70S ribosomes is 2:1, whereas the RNA protein ratio in eukaryotic, that is 80S ribosomes, is 1:1. Coming to the chemical composition of ribosomes, ribosomes contains mainly RNA, proteins, enzymes and metal ions. What is this ribosomal RNA? Ribosomal RNA that is present in ribosomes are called as ribosomal RNA. Already we have seen that in eukaryotic cells it has 28S rRNA, 5.8S rRNA, 5S rRNA and 18S rRNA are found. Whereas in prokaryotes it is 23S rRNA, 5S rRNA and 16S rRNA are found. Apart from that, it contains 50 to 80 proteins, that is ribosomal proteins. What about the enzyme machinery? Enzymes, the ribosomal enzymes play important role in protein synthesis. They are grouped into three types, namely initiation factors, elongation factors and termination factors. Apart from all this chemical composition, it also contains various metal ions. Ribosomes contain number of metal ions such as magnesium, calcium, manganese and iron. What are the main functions of ribosomes? As already I have mentioned that ribosomes mainly involved in protein biosynthesis. So first function is it assembles amino acids to form proteins that are essential to carry out cellular functions. We all know that protein is the building blocks of our body. So the mainly ribosomes helps in the protein biosynthesis. How it is produced? The DNA produces mRNA by the process of DNA transcription process, where the mRNA is synthesized in the nucleus and it is transported to the cytoplasm for the process of protein synthesis. The ribosomal subunits in the cytoplasm are bound around the mRNA polymers. Then the tRNA synthesize these proteins. So hence ribosomes is the main site for protein biosynthesis. Here the protein that are synthesized in the cytoplasm are utilized in the cytoplasm itself, so that the protein synthesized by bound ribosomes are transported outside the cell. Thank you for your patient hearing. We shall meet in another lecture.